Hello, my dear students. This is your host, Dr. Syed Parker. Today's class, let us study about chromatography and electrophoresis. Moving on to the chromatography, it is one of the versatile separation technique used to separate and analyze the complex mixture based on their differential migration of components in different phases such as mobile and stationary phase. The principle of chromatography relies on the distribution of components between the mobile phase and stationary phase. Let us see the overview of how it works. There are different types of chromatography. Here you can see gas chromatography, paper chromatography, column chromatography, etc. Every technique has its own advantage and as well as disadvantage. But the motive of all the technique is to separate the mixtures in a given sample. The principle here is basically the separation or the distribution between two phases. It might one phase which will be always traveling, it is called as mobile phase, and the other phase is stationary phase. In mobile phase it might be liquid solvent or gas. The stationary phase is always an adsorbent where the components get separated while it passes through the stationary phase. If we see column chromatography as an example, here you can see this is the stationary phase in a column and you have loaded your sample. Once you add your mobile phase onto the sample starts migrating. Here you can see it starts migrating. Here the weaker interaction or absorption with the stationary phase results in faster movement, whereas stronger interaction or adsorption, here the separation is slowly. So each fractions get separated at different time interval, and this separation is collected, which we call it as eluting sample or eluting molecules. Each separated sample is collected in a different vials or test tubes and analyzed for the separated compounds. Similar principle is followed in the TLC, but in a reverse order where the solvent migrates from bottom to upper direction. Apart from this, you have a gas chromatography. Here, the mobile phase is gas and the stationary phase might be liquid or solid. Here, it is widely used in separating volatile compounds such as organic molecules and gases. In liquid chromatography, as I discussed, there are different types, and here the mobile phase is liquid and the stationary phase is a solid medium. Under this, you can see high performance liquid chromatography, which is nothing but the HPLC, and thin layer chromatography. Apart from this, you also have ion exchange chromatography, size exclusion chromatography, affinity based chromatography. Let us see one by one. In ion exchange chromatography, here you can see there are different positively charged protein binds to a negatively charged bead. The beads present inside the column or the chamber contains negatively charged, and the positive charge present in your mixture goes and binds to the negative charge beads. Here you can see the working principle. You have different beads. This might be positive or negative. Here we are seeing the negative charge. Once the mixture is poured inside this column, then the positively charged get attracted to the negatively charged beads and the remaining positive charge present in the mixture gets eluted. This is based on ion exchange chromatography. Whereas here, this based on the size, you can see here based on the size of the molecules or particles. Once you add based on the size, it's start separating. The smaller particles tend to separate at the last, whereas the medium particles, and the particle with more like larger size try to get separated faster, and the retain based on the retention time you can record the separation. Here you can see the bigger particle are eluting first, followed by the medium size particle, then the smaller size particle. Moving on to the affinity based chromatography. Here you have the expressions on the beads, and these beads having a specific expression, and the components or the mixture with that specific size goes and binds to this expressions. Here you can see, and the remaining stats get eluting. Here you can see the triangle shaped particle are getting bound to this expression, and other particles such as spherical, square, rectangle, and oval shaped particles are getting eluted. Same things happen, and this eluted components are collected and investigated further. So moving on to the applicative properties, application of chromatography. Why chromatography is so important in modern sciences? Almost every industry, you may take pharmaceutical industry, chromatography techniques are followed in order to check or identify or discover a new compounds bearing biological activity, drug related activity. Similarly, in environmental monitoring, analysis of pollutants in air, water, and gases, air, water, and soil are carried out using chromatography. Also, biochemical and biotechnological application is also applicable when it comes to chromatography. Food and beverages industries also uses chromatography in order to detect the contaminants and desired or flavored compounds. Apart from this, forensic science, petroleum, clinical diagnostic kits, and material science also use chromatographic techniques. Now, moving on to the second section of this session, electrophoretic techniques. Electrophoresis is nothing but a technique or a movement of charged particle under the influence of electric current. The movement depends on the size, shape, and type and the charge of the particle. In order to, whereas in the chromatography the separation was based on the liquid and solid phase, whereas here in electrophoretic technique the separation is based on the electric charge, and this electrophoretic technique is carried out in a specific instrument, and each instrument has its own application in order to carry out electrophoresis, and it is carried out in buffer solution which maintains the electric conductivity constantly. So here you can see the particles with positive charge goes and binds or gets attracted to the negative charge of the instrument, whereas the particle with the negative charge goes and binds to the component positive charge component in the instruments. And this is an agarose gel, and this agarose gel is created wells, and inside this well we add the mixture which needs to be separated. The electrophoretic technique is usually used to separate the biomolecules, especially DNA, RNA, and proteins. DNA and RNA are the net charge is negative charge, whereas the protein might be positive or negative depending upon the type of amino acids. What we usually do, this is the chamber here. We create agarose gel by dissolving 0.7% of agarose, and this agarose is placed into a chamber, and we create wells. Here you can see the different wells using a comb. We create this, and once this wells are created, we place this into the electrophoretic unit. which is filled with buffer. And this, once we add the mixture and apply the electricity, it starts migrating. Here you can see this provides the electricity, and here it starts migrating. Once it is done, we disconnect the electricity and remove the agarose gel carefully, and this, and remove the pollution. And once the buffer solution everything is removed, we take this for further analysis. And here you can see the solution added to this well after the separation. You can see the different bands, and different bands are observed using the software. So moving on to the components of electrophoretic techniques. Electrophoresis chamber, which we have already seen. Electrodes, anode positive electrode and cathode negative electrode, which are immersed in buffer solution, creating the electric field. And negatively charged particle goes and attracts to anode, and positively charged particles get attract to cathode in electrophoresis technique. The supporting media here or medium here is agarose gel, polyacrylamide gel, or a capillary tube. The buffer solution, as I discussed, it provides the ions for conductivity and controls the pH. And wells, we have already seen the wells and loading buffer. The working principle, which we discussed, based on the charged particle, it gets separated, and this separation also be influenced with the size, shape, and type of the particle. Apart from this, we have different types of electrophoresis. Capillary electrophoresis, and in capillary electrophoresis, separation of ions or molecules takes place in the capillary tube filled with electrolyte solution. And protein electrophoresis, we have protein electrophoresis. Here we use SDS phase for separation of protein, and this type of protein SDS phase separation results in also identification of protein molecular weight, and also we can purify a desired protein using this technique. Along with this, you have immunoelectrophoresis, paper gel electrophoresis, pulse field gel electrophoresis. And in pulse field electrophoresis, it is used to check the size of entire chromosomes. And in paper gel electrophoresis, it is usually practiced for body fluids and serum. When it comes to application part, DNA fingerprinting, protein analysis, PCR product analysis, genetic testing, etc. are some of the application. So with this, students, let me conclude this session where we have discussed about the chromatographic techniques as well as gel electrophoresis. The chromatographic techniques you have already practiced during your practical sessions, and we have practiced TLC. I hope you remember. If you have any doubt, please don't forget to call me. I wish you very good for your upcoming exam. Goodbye.